My name is Paul Michaels. I am with Biospherics. Biospherics, we are located in upstate New York, just north of Syracuse, New York, in the United States. And this is a presentation that was given at the GSCN conference in Jena, in case you missed it. So the title of my talk is Avoid Irrelevant, Non-Reproducible Data, the Neglected Variable, Physiologic Oxygen. So let's get started. So the scientific community has relied on animal models for a lot of their data. There are many problems with the animal model, but the three main problems that we face with the animal model is: one, they're enormously expensive. Two, there's always ethical issues being brought up about the animal models, and three, quite frankly, at the end of the day, they're simply not human models. So there's been an ever-increased reliance on cell-based models. But for cell-based models to be effective, they need to be relevant and they need to be reproducible. So let's take a look at the first one, irrelevant data. So we simply have cell-based models that do not predict, and it creates this gap of irrelevant data, and this gap is what needs to be closed for the cell-based models to have the predictive capabilities that we need to show. So when it comes to irrelevant data, I look at it like a dartboard. I look at this gap as a dartboard. So if we're shooting for our goal or target here, but all of our targets are over here, the data is simply not relevant. And what are the downsides of that? Well, there's many, and I'm not going to go through all of this, but industry metrics have bringing a single drug to market can cost anywhere from $800 million to $4+ billion for a single drug, and that's with an average time of 8 to 12 years to bring this drug to market. Not to mention the fact that if a drug is withdrawn, it's hundreds of millions of dollars in cost to withdraw that drug. And for those drugs, those small amount of drugs that do make it to market, about 10% of those drugs require added health and safety issues added to their package insert. And that's not to mention the litigation costs paid out to victims on recalled drugs can be in the multi-billions of dollars for a withdrawn drug. So Dr. Harvath and colleagues did a review looking at the causes of irrelevant data, and these top 7 have been addressed, or significant improvements have been made on these top 7, with the exception of one cause of irrelevant data, and that is the appropriate atmospheric conditions. And this is even if and this includes controlling for oxygen. In fact, they spend a significant amount of time in this paper talking about the importance of physiologic oxygen control as it relates to irrelevant data, along with the other factors for irrelevant data. So if you look at the cell culture history over the last 100+ years, and if you notice the top 4 or 5 here, actually not only do they mimic, they're close to, or they exactly mimic what Harvath and group had demonstrated in some of the causes as well. But if you look at this, there's been significant improvements in the technology over the years when it comes to cell culture history, except for one area, and that is the incubation and handling environment. It has remained the same for over 100 years. So is oxygen concentration important? Well, let's take a look. So over the last 50 years, we found that oxygen plays multiple roles, and because of this multiple role configuration of oxygen, it's complex, but we found that oxygen plays roles in electron sink, redox, enzymatic reactions, and more recently in gene expression or phenotype. So oxygen plays, or is a central factor in many basic cell functions, whether that's redox, whether that's metabolic function, or gene expression, and more. It is a central factor in all of these. So if your cells could pick an atmosphere, what would it look like? Well, obviously they're going to pick the temperature at 37 degrees Celsius and the carbon dioxide at 5%. But what is the appropriate physiologic oxygen concentration for the cells? Well, that really depends on location. Again, it depends on what cells you're studying and where they're located in the body. As you can tell from this graphic, there's a large gradient in terms of physiologic oxygen concentrations. If you look at the lung, you're talking around 10 to 13% for oxygen concentrations, down to cartilage, which it can be at 0 or 1%, and everywhere in between. Even in the lungs, when you're talking about the lung cells, nowhere in the body, including the lungs, are cells exposed to 21% or room air oxygen. So whether your cells are in the incubator, whether they're in the hood, or whether they're in the machine, your cells are never exposed to room air oxygen when they're in the body. So I can talk about all this, all is well and good, but here's a real live example. Dr. Ian Mudway and colleagues published a paper called Quantifying the Magnitude of the Oxygen Artifact Inherent in Culturing Airway Cells Under Atmospheric Oxygen, room air, 21%, versus physiologic levels. And what they found were cells that were cultured under room air, the cells pre-adapt to the room air, which alters their redox capacity, which reduces their predictive power. But more importantly, what they found were cells that were cultured under physiologic oxygen levels. They found that not only were the results different compared to the cells cultured under room air, but they found that they matched what they saw in vivo. So basically, their predictive power was much more effective. So I think we can all agree that culturing cells under room air oxygen is irrelevant at best. No one in the cell culture method says, let's carry out these steps in non-physiologic, irrelevant room air throughout the entire process, but yet that is exactly what happens in the traditional cell culture labs that we do every single day. So I hear this probably at least once a day, if not 2 or 3 times a day. Well, my cells are only out of the incubator for a few minutes. Does it really matter? Well, it does. And Pusiger and colleagues in 2006 established that HIF-1 alpha in well-oxygenated cells, 21%, displays one of the shortest half-lives, less than 5 minutes among cellular proteins. And more recently, Broxmeyer and colleagues found that cells exposed to room air, even short exposures, less than 15 minutes, showed a significant increase in ROS production, which actually mimicked ischemic reperfusion injury. The cells were being damaged, and not only were the cells damaged, and not only was the damage significant, they found that it was irreversible. The cells could not be saved, and they called this EPHOS, or extra physiologic oxygen shock and stress. So I bring it back to my cells are only out of the incubator for a short period of time. Does it matter? Well, it does very much matter. And again, I bring this back to our problem number 2 in terms of reproducibility, to the dartboard. So not only are we not hitting the target, we're not even getting a good grouping around the target in most of our cell-based models. And Dr. Leonard Freeman and colleagues published a perspective on the economics of reproducibility when it came to scientific research. And they found that not only was it a huge economic burden when they're talking about $28 billion, and this is only U.S. figures, $28 billion a year is wasted in research publications that are not reproducible. But they go on to say that on top of the economic burden, it delays our advancement of knowledge in disease states, and it delays our advancement in cures for patients that are suffering from diseases such as diabetes or Alzheimer's or cancer or diabetics, what have you. It's an economic burden on top of delaying the advancement of our knowledge as well. And Dr. Freeman and colleagues go on to say that the causes of irreproducibility are complex, ranging from resource limitations to pressure to publish to variable journal policies, but the one underlying factor that links all the causes of irreproducibility together is the lack of or no quality systems. And when it comes to a scientific paper, the sources of variation are many. Whether that's in the same lab, whether that's in a nearby collaborating lab, or in a distant lab. There's many sources of variation, but the one constant and significant cause or source of the variation resides in the cell conditions, what the cells are exposed to. And the one good thing about this is the fact that cell conditions is one of the few areas, if the only area, that is still under your control. So think about your traditional laboratory, and let's say, just for the purposes of this talk, that you are controlling for oxygen in your incubator. The only time your cells are under optimal conditions in the conventional lab is when the cells are inside the incubator, the door is shut, and all of those set points are reached. So the carbon dioxide is at 5%, temperature is at 37 degrees Celsius, and the whatever oxygen tension you're studying is reached. But as soon as that door of that incubator is open, your cells are exposed to the room air in suboptimal conditions. So O2 and pH levels increase, CO2 levels and temperature decrease, and this happens each and every time that door of that incubator is open, and that simply creates variability in the data. And again, the conventional hood, as soon as those cells are removed from the incubator and brought over to the BSC, the cells are subjected to suboptimal conditions in room air. Again, O2 and pH levels are increase, CO2 and temperature decrease. And think about this. One technician can do the manipulations or the processing of the cells in 10 minutes, while another technician to do the exact same manipulations takes 15 to 20 minutes to do the same processing. Think, what happens if they get a phone call? What happens if they have to go to the bathroom? What happens if they get a text or a tweet? God forbid we miss a text or a tweet these days. That would be unthinkable. But this all relates back to variability created in the data. And I bring this slide back, Dr. Leonard Freeman and colleagues, just to remind you that the one factor that links all the causes of irreproducibility together is the lack of or no quality systems. So every manufacturer has quality principles in place, whether that's the automobile industry, whether that's the cell phone industry, or what have you. Every manufacturer has quality principles in place. They're there to improve the efficiency, but more importantly, the quality of the product. So what are the live cell industry quality principles? Well, quite frankly, there is none or very little when it comes to quality principles. So again, bringing this back to cell-based models, to be effective, they need to be relevant and reproducible. What do the cells need from us? Well, they need a set of quality principles. The cells need physiologic simulation, they need full-time optimization, full-time protection, unique protocol, and dynamics. For purposes of this talk today and the time we have, I'm going to focus on the top two, physiologic simulation and full-time optimization. So what I propose is a set of quality principles for the live cell industry to be called cytocentric, and that is putting the needs of the cells before the needs of the people. So physiologic simulation, unbroken physiologic conditions. Think of your traditional lab. I call it a people-centric lab because it's been designed to meet the needs of the people. But what do you see in this picture? You see chairs and desks and shelves, and yes, there's incubators and there's hoods. It looks like microscopes and other machines, but it is a lab built around the needs of the people. So, and this should be a familiar sight. This is a conventional incubator opening up into the room air, exposing the cells to suboptimal conditions as soon as that door of that incubator is opened. And again, yes, there's some added protection when it comes to the BSC with the laminar flow, but the whole time your cells are exposed to the room air in suboptimal conditions. So a conventional incubator, and let's say you are controlling for oxygen, you have control over basically three parameters: temperature, carbon dioxide, and oxygen. What does a cytocentric incubator look like? Well, a cytocentric incubator looks like this. And this is the incubator in the back. The door is open for purposes of this picture. But basically, we've extended the incubator out into a closed aseptic processing hood. So cytocentric incubators have control over those three parameters: oxygen, CO2, and temperature, but they have control over many other gases as well, but they only open into a controlled aseptic environment. And again, there are no physiologic atmospheric controls on a conventional hood or BSC. What does a cytocentric hood look like? Well, we've already seen it, but this is the cytocentric hood. So the cells, these are the incubators here, these black boxes. The cells, the incubators open into a closed aseptic hood. So the cells are cultured and processed all under the same atmospheric conditions. And each of these black boxes here in the back are independently controlled incubators to run multiple different protocols if necessary. And we understand that, your process or your methods consist of more than just incubation and processing under a BSC. A lot of times your methods or your process include going to a microscope or multiple different machines. And Biospherics has become very good at being able to manage and enclose third-party pieces of equipment. We're all familiar with this. This is the Seahorse Analyzer. This is a plate reader. This is actually a CS plate reader. And we've gotten good at any piece of third-party equipment pretty much that you can think of we've enclosed, such as a bioreactor. And I show this picture not to scare you, but just to show you that whether it's large or small, we have the capability of enclosing and managing these machines to keep your cells exposed to only those atmospheric conditions that you want them exposed to. Obviously, a machine like this or any machine produces, one, it produces heat, but two, it produces particles. Biospherics has gotten very good at being able to manage those particle producing machines and heat producing machines to keep those cells all under the same atmospheric conditions throughout the entire process. This machine is a large TCAN filling system that is at the Cleveland Clinic that runs cell-based assays. And again, I put this up here just to show you that large or small, we have the capability of enclosing any third-party piece of equipment. So moving on to full-time optimization. Again, bringing you back to your conventional lab, nowhere in a conventional lab are your cells under full-time optimization, even if your cells are in the incubator, simply because as soon as that door of that incubator is opened, again, the cells are exposed to suboptimal conditions. So think of it like this. This is a people-centric lab where you have your incubators, your closed hoods, your BSCs, microscopes, centrifuge, what have you, any third-party pieces of equipment. Anything you can do in a people-centric lab, you can do in a cytocentric lab. We can enclose, or Biospherics can enclose your entire process. So your incubators are here in the back, opening up into a closed aseptic hood, along with your microscope and centrifuge here. We can enclose your entire process where your cells are cultured and processed and machined all under the same environment from start to finish. So again, this is an example of a cytocentric lab where you have your processing chambers, incubation, and third-party pieces of equipment. This is a general small, this is a system that resides in our home office where our CSO does a lot of her data gathering in this machine. But basically, the cytocentric laboratory protects your entire workflow from start to finish, where your cells are basically under the same atmospheric conditions from start to finish. So this is all well and good. I can talk about this all day long, but I want to give you a real world example where an institution has gone to this cytocentric approach. Dr. Philip Schwartz and colleagues published a paper titled Culturing Human Pluripotent and Neural Stem Cells in an Enclosed Cell Culture System for Basic and Preclinical Research. And this is on Jove, by the way, so you can actually go and see them working in this closed cell culture system. But I'm going to do what I normally wouldn't do in a presentation, but I'm going to read directly from the abstract. And the reason I do that is, one, I can't say it any better than Dr. Schwartz does in this abstract, and two, he basically outlines almost every single point that I go through in this presentation, in this abstract. So the abstract starts off saying, this paper describes how to use a custom manufactured, commercially available enclosed cell culture system for basic and preclinical research. Biosafety cabinets and incubators have long been the standard for culturing and expanding cell lines for basic and preclinical research. It goes on to say, as the focus of many stem cell laboratories shifts from basic research to clinical translation, additional requirements are needed of the cell culturing system. All processes must be well documented and have exceptional requirements for sterility and reproducibility. In traditional incubators, gas concentrations and temperatures widely fluctuate anytime the cells are removed for feeding, passaging, or other manipulations. Such interruptions contribute to an environment that is not standard for GMP or GLP guidelines. And it finally goes on to say the motivation to move from the standard BSC and incubator system to a closed system is that such interruptions can be made negligible. Closed systems provide a workspace to feed and manipulate cell cultures and maintain them in a controlled environment where temperature and gas concentrations are consistent. So again, I can't say it any better than Dr. Schwartz does in the abstract. And again, he points out almost every single point that I'm trying to make in this presentation in the abstract, and that's why I read it. So with that, I'd like to finish and thank you for listening. If you have any questions, I would be more than happy to set up a time that we can talk and just go over different solutions that many of our customers have found extremely helpful in their research. My email is right here below. Please reach out to me at any time. And again, with any questions, I'd be more than happy to talk over the phone or communicate over email. So thank you very much.